Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 6 from our Genetics Essentials, Concepts and Connections 4th edition textbook. This chapter deals with chromosome variation, so let's get started. Now, we touched on this before, but recall that chromosomes have different morphology. They could have the centromere located near the center of the chromosome so that you end up with two arms of equal length. You could have what is known as submetacentric centromeres, where the centromere is displaced towards one end, creating a long arm which is denoted Q, and a short arm which is denoted P. So when you see Q, you know that's referring to the long arm of the chromosome, and when you see P, that denotes the short arm of the chromosome. Don't forget there's also what's known as acrocentric chromosomes, where the centromere is near one end, producing a long arm and a knob or satellite at the other end. So it's really pronounced long arm at one end and a stubby knob at the other. And then you have telocentric, where the centromere is at or very near the end of the chromosome. Don't forget that the ends of your chromosomes are called the telomeres. The very ends of the eukaryotic chromosomes are called the telomeres. So just a little bit of review about what we've learned previously. And here we're looking at a karyotype. What is a karyotype? The karyotype is a complete set of the chromosomes possessed by an organism. So this is a karyotype of a human. Notice that there are 23 different chromosomes, and because we have two homologous sets of chromosomes, we have two chromosome 1s, two chromosome 2s, two chromosome 3s, two chromosome 4s, so on and so forth. And our final set of the 23rd set are known as the sex chromosomes. We touched on that in the last chapter, where the X chromosome, it's much longer, has a lot more useful DNA on it. Then our Y chromosome is much shorter, stubbier, and remember the main gene on there of interest usually is the SRY gene. Now, notice how the karyotypes are usually arranged from longest to shortest chromosomes. So chromosome 1 tends to be the longest, chromosome 22 the shortest, and of course you have the shortest one technically is the Y chromosome, but the X chromosome's longer. So this is a karyotype of a human, and you could tell this would be from a male because they have the X and Y chromosomes here as the 23rd chromosome. Now, if it was a female, you would see two X chromosomes, and if it's a male, you see the X and Y chromosomes. Now, in your textbooks, you may see images like this. This is called banding. Mitotic chromosomes are stained so that it reveals the banding. So why do you want to stain? Well, it's nice to be able to stain mitotic chromosomes to see either AT-rich regions. This here is called G-banding. It's a special type of staining technique that reveals AT, adenine thymine rich areas of the karyotype. There's also another type of staining that highlights the AT-CG differences a little bit better. That's called Q banding. You could have a type of staining that highlights the centromere, the position of the centromere, and that is called C banding. C banding. And then you have another type of staining technique that reveals banding highlighting the C and G rich areas, and that's called R banding here. So again, when you look in your textbook and you see these banding patterns, that's what revealed the banding patterns, was this staining technique. And remember, it's called a mitotic chromosome because the chromosomes are as they would be during mitosis. They are basically arrested. Usually when you make a karyotype, you arrest the cell cycle in metaphase, and during metaphase the chromosomes have condensed fully into these X shapes, right? So you can see those mitotic chromosomes there. Now, when we're talking about chromosomal mutations, there are different consequences and different types of mutations as well. We're going to get into those, but these are very important in evolution. They are important in agriculture, and we're going to touch on that. Now, what you can have is rearrangement of the chromosome, where the structure of the chromosome itself is altered. You could have what's known as aneuploidy. This means that you have one or more individual chromosomes added or deleted. So if you have an extra chromosome, or if you're missing a chromosome, or if you have two extra chromosomes, something like that. And then polyploidy, which means that you have a whole extra set of chromosomes. So instead of having, for example, 2N, two homologous sets, one set from your mom and one set from your dad, you may have a whole extra set of 23 chromosomes, for instance. That's the example there. So you could be 3N or 4N or 5N or more. You know, you could get up to 8N, I've seen, for strawberries. Now, you can see here the difference between those three. You can have, again, chromosomal rearrangements. This changes the actual chromosome structure. So in this case, it looks like alleles C, D, and E have been duplicated. So this part of the chromosome has been duplicated. So now you have two copies of that part of the chromosome. See how that would extend the chromosome out and change the structure of this chromosome. Here's aneuploidy. Aneuploidy, you have, one or more chromosomes in the cell or missing from a cell. In this case, it looks like it has one extra chromosome. And then you have polyploidy, where you have a whole extra set of chromosomes. So you have a whole extra set. You have a third one of this chromosome, a third one of this chromosome, a third one of this chromosome. So the ploidy has gone from 2N to 3N. Now, what type of rearrangements are there? So we talked about chromosome rearrangements. What kinds of rearrangements are there? So we're going to touch on them, but there's duplications, which this is an example here of a duplication of these alleles C, D, and E. That part of the chromosome was duplicated. There's deletions, where you could actually lose part of the chromosome. Inversions, where part of the chromosome flips around. And translocation, where part of a chromosome moves to another chromosome. So here's duplication, a graphic design of duplication. You have your chromosome with alleles A, B, C, D, E, F, G. And what happens is, on that same chromosome, there's a duplication event of this region of the chromosome. So this region of the chromosome is copied, and now you have two copies of that part of the chromosome next to each other. So now you have two of the E allele on this chromosome, and you have two of the F allele on this chromosome. So now you have twice as many of the gene of interest, E and F. You have twice as many gene E and F on this chromosome, and this chromosome has gained in length as well. In a deletion, the opposite is true. You've actually lost this part of the chromosome. That part of the chromosome got deleted. So you have A, B, C, D, G. You've lost E and F altogether, so you've lost that genetic information. And a lot of times, in case you're wondering, in a textbook or something, you'll see the sign delta, the Greek letter delta, the triangle, and that will denote a deletion. So if you ever see like CCR5 delta 32, which is the mutation that allows some people of European ancestry to become immune to certain strains of HIV, you see CCR3 CCR5 delta 32, the delta would mean that there is a deletion in that part of the chromosome, in that particular area. Now, what is an inversion? An inversion is where part of the chromosome is actually flipped around, inverted, in the same chromosome. So imagine if I had scissors and I cut here, flip this around. So the alleles D, E, F are now going the opposite way. And this can actually cause damage to the chromosome, because you could actually disrupt a gene. What if the inversion happened in the middle of the allele F? Well, then you would destroy the F gene. Or what if it happened in the middle of the allele D? Then you would actually destroy the D allele. And translocation. So translocation can either be you take part of the chromosome and you can move it to another chromosome. It can either be reciprocal, where part of that chromosome is swapped, or it could be one way, where you're only giving information to another chromosome. So it can either be reciprocal or non-reciprocal. In reciprocal, one chromosome exchanges information with a non-homologous, that's the key part, a non-homologous chromosome. In non-reciprocal, a chromosome gives information to a non-homologous chromosome. So this chromosome here with alleles A through G is a different chromosome and not the same chromosome number. It's not a homologous chromosome to this chromosome, which has alleles M through S, if that makes sense. All right, so to understand why such mutations or rearrangements might be impactful to an organism, you have to understand how alleles work to begin with. So take a look here. Here's a gene with alleles A, B, and C. The alleles A, B, and C will produce gene products. These are proteins, such as enzymes. These proteins could be transcription factors. These proteins have jobs in the cell. And you have to have interaction of these gene products, these proteins. And they have some function during normal development. Does that make sense? If you have too many copies, as in, let's say, a duplication event, if you have too many copies of allele B, you're going to end up with too much of B's gene product, too much protein, right? And that protein might have a job, and because it has now a incorrect ratio of this protein in the cell, you have excess protein B from allele B, that might actually cause abnormal development. Does that make sense? So if you have a duplication, you're going to have too many copies of allele B, which will give you too much protein B, too much protein from allele B, and that might cause developmental defects. What about a deletion? What if I deleted allele B? What if I deleted allele B? Well, then I would have too little of allele B in the cell, and I might have abnormal development. What if there's an inversion? Like I said, an inversion might actually destroy B, because what if the inversion started at the middle of allele B? So you can see how chromosomal rearrangements might have deleterious effects for normal development for the creature. Let's do a concept check real quick. Chromosome duplications often result in abnormal phenotypes because, we just touched on this. Let's see, A, developmental processes depend on relative amounts of proteins encoded by different genes. Yes, it's A, right? So you need, like I said, in your cells, you have homologous chromosomes. So you have allele A on this chromosome from dad, and you have allele A on the homologous chromosome from mom. If you have extra copies of allele A, or if you're missing copies of allele A, that's going to result in different amounts of the protein from that gene, and that can result in developmental defects. Does that make sense? Here's just a a list of examples of what we know already about how duplications, chromosomal duplications, chromosomal deletions, and how these affect human development. And you can see there's a myriad of different human symptoms that can arise from various duplications or deletions. And again, why is that the case? Well, because you're changing the amount of gene expression, the amount of protein in the cell when you have these duplications or deletions, and that's going to affect normal development and function. So again, what about deletions? You have loss of chromosomal segments. The effects of deletions, well, again, you'll have imbalances in gene product. You might need 2 copies of that gene for ideal function and development of the embryo. You might have what's known as haploinsufficiency, which is basically the same concept. Haploinsufficiency, where 2 copies may be necessary for gene function, or the expression of the normally recessive gene would persist. This one's interesting. What if you delete the dominant allele on chromosome 1? What if you delete the dominant allele? Well, then on the homologous chromosome, the recessive allele might express itself. This is a concept called pseudodominance. You guys follow? So again, you have mom and dad's chromosome 1. Dad's chromosome 1 has a wild type allele. Mom's chromosome 1 has a mutant disease allele. What if I delete dad's chromosome 1 so I lose that wild type allele? Well, all that's left is mom's chromosome 1 with its mutant disease allele, and I'm going to express that disease phenotype. Does that make sense? Because you've now just deleted the wild type allele from the wild type from the homolog. Concept check number 2. What is pseudodominance and how is it produced? By chromosome deletion. We just explained that. If you delete a wild type gene, a dominant wild type gene, if you delete a dominant wild type gene from one homolog, well, then a recessive disease allele, or a recessive allele in general, from the other homologous chromosome that was not deleted is going to express itself. So again, pseudodominance, let's see what their definition is. Pseudodominance is the expression of a normally recessive mutation that is produced when the dominant wild type allele in a heterozygous individual is absent due to deletion on that chromosome, on that one chromosome. What about inversions? We talked about inversions. In inversion, DNA is neither lost nor gained. Does that make sense? DNA is neither lost nor gained. It's just flipped around, right? DNA is flipped around. But this can still have a bad effect on development. What if it cuts what if it breaks a gene apart? Like I said, what if the area where the chromosomes cut to flip around, what if you cut right in between a gene? What if you destroyed a gene in the process of flipping that chromosome around? Well, then you're going to have what's known as position effect. You're going to have a break in that gene. Position effect is a different thing. Let me explain a position effect. If you flip chromosome around, well, then you can have what's known as position effect. So, for example, that part of the chromosome might get sequestered because of histone proteins. That part of the chromosome might not be accessible to enhancers or promoters. That part of the protein might not, that part of the chromosome might not be as easily read by the machinery of the cell. Does that make sense? So again, the 2 reasons inversions are bad. One is you can break a gene apart at the points where you broke the chromosome to flip it. 2 is position effect, meaning a gene might be perfectly expressed just fine on this part of the chromosome, but you flip it and put it on another part of the chromosome. Well, that part of the chromosome might not be as easily read because it could be, like I said, a part of the chromosome that forms heterochromatin and it gets sequestered so that you can't actually read that information, etc. That's position effect. Now, here's an interesting thing. There are two types of inversion that you need to know about. Depending on if it involves the centromere or not. If you flip the chromosome and that includes the centromere, that's called a pericentric inversion. Inversion that includes the centromere. If you have a inversion and that does not include the centromere, that's somewhere, let's say on the long arm or on the short arm, well then that's a paracentric inversion. So you can see here, here's a picture of an inversion. You can see that the inversion occurred as pericentric because the centromere was involved in this inversion. Now, if the inversion had gone on over here, well then that would be a paracentric inversion. Now, here's an interesting fact. Remember, I told you that chromosomal mutations in general are important for evolution. Well, it's shown that with G-banding has shown that several human chromosomes differ from those of chimpanzees by only a pericentric inversion. So, for example, our human chromosome number 4 is only different from chimpanzee chromosome number 4 by a pericentric inversion. Isn't that fascinating? Translocations. Remember, translocations entail movement of genetic material between non-homologous chromosomes, so from chromosome 3 to chromosome 5, for instance, or within the same chromosome. You could also translocate within the same chromosome. Now, non-reciprocal translocation, we touched on this before. This is when genetic material moves from one chromosome to another without any reciprocal exchange. So I'm taking part of chromosome 3 and I'm putting onto chromosome 5, but none of chromosome 5 is going the other way. None of chromosome 5 is reciprocating to chromosome 3. That's a non-reciprocal translocation. Reciprocal translocation, here's an example. It's a two-way exchange. You're taking part of chromosome 3, sticking it on chromosome 5, part of chromosome 5 onto chromosome 3, and so you're exchanging information that way. And then you have a Robertsonian translocation. This is a special type. Long arms of 2 acrocentric chromosomes become joined to form a common centromere through a translocation, generating a metacentric chromosome with 2 long, usually very long, and 2 very short arms. Frequently leads to deletions. I'm going to show you an example of Robertsonian translocation. Here it is. Robertsonian translocation. You've got, here's a chromosome, chromosome 1. Here's a different chromosome, chromosome 2. Here's what's happening. You're going to have translocation between the two, but it's going to result in the long arm of this chromosome moving to the long arm of the other chromosome. So you end up with one really long chromosome with 2 long arms. And then you also result in this nubby, tiny chromosome as well, this fragment chromosome. And this is often lost. This usually fails to segregate and is often lost, leading to a deletion there. So what is the outcome of a Robertsonian translocation? Concept check. 2 acrocentric chromosomes? Nope. 1 metacentric chromosome and 1 chromosome with 2 very short arms? Yes. That's exactly right. Isn't that what we saw here? 1 metacentric chromosome and 1 with 2 tiny arms that's usually lost. That's right. So now let's move on from chromosomal rearrangements to the concept of aneuploidy and polyploidy. So variations in copy number, in the number of chromosomes, is aneuploidy or polyploidy. So let's talk about aneuploidy first, because remember that was the addition of 1 or 2 or more chromosomes, or the deletion of 1 or 2 or more chromosomes. So what are the causes of aneuploidy? How could you end up with an extra chromosome? There are three main ways. Deletion of centromere, like if you delete a centromere during a chromosomal rearrangement. Robertsonian translocation, remember that short nubby chromosome that results during a Robertsonian translocation, that could be lost. Or nondisjunction, this is actually quite common, nondisjunction during meiosis or mitosis. That's how you could end up with aneuploidy. Here's some terminology while we're talking about aneuploidy. Nullisomy would be if you lose both chromosomes. So now you've got 2n minus 2. So for example, what if I lost both my chromosome 1s, right? Then I'd have 0 chromosome 1s. So it's denoted like this, 2n minus 2, because I've lost both chromosome 1s. Monosomy, this is loss of a single chromosome. So I've lost one chromosome. Trisomy, I've gained a single chromosome. Tetrasomy, I've gained 2 homologous chromosomes. I've gained 2 chromosomes. So how does this result? Again, I said the main way you end up with aneuploidy is what a process known as nondisjunction. Think of nondisjunction as anaphase gone wrong, right? Remember what happens during anaphase? You have the chromosomes lined up at the center of the cell during metaphase, and then during anaphase, it's the job of the cell and those spindle, remember those spindle proteins and the centromeres and the kinetochore and all that? Remember, the job during metaphase is to line up chromosomes. The job of anaphase is what? The job of anaphase is to separate the chromosomes evenly in the cell. Remember mitosis, meiosis, meiosis 1, meiosis 2, all of those have disjunction. So you can think of disjunction as chromosomes line up during metaphase and separate during anaphase. Well, what if this? Think about it. What if chromosomes lined up during metaphase and they didn't do the separation correctly, so you ended up with both of these going to one cell? Think about it. Wouldn't that cause aneuploidy? Too many chromosomes in this cell over here and too few chromosomes in the other cell over there? That's exactly right. That's exactly what nondisjunction is. Think of metaphase and anaphase not going correctly. You wanted to split the chromosomes a certain way, and they didn't split that way. That's the simplest definition of nondisjunction. So this can happen during meiosis 1. Remember, during meiosis 1, you've got metaphase 1 and anaphase 1. Well, remember what lines up during meiosis 1? The tetrads line up. And then what if the whole tetrad goes to one cell and none of the tetrad goes to the other cell? Well, then you would end up with, for example, no chromosome 1 in this cell, 4 chromosome 1s in this cell. Does that make sense? No chromosome 1 in this cell. All 4 chromosome 1s went to this cell. So then in meiosis 2, these two cells have no chromosome 1. These two cells have these two cells have 4 chromosome 1s. So when the sperm comes along, let's say this is the sperm from the other parent, there's fertilization. These two zygotes, these two embryos, right? These two zygotes have 3 chromosome 1s, 3 chromosome 1s. So that's 2n plus 1. That's a trisomeric, or trisomic, I should say. And then you have these two gametes getting fertilized, and they only have 1 chromosome 1. Does that make sense? 1 chromosome 1. So this would be a 2n minus 1. So one way of getting aneuploidy is nondisjunction during meiosis 1, where the tetrads do not separate correctly. The homologous pairs do not separate during meiosis 1. Or what about meiosis 2? Remember meiosis 2? So let's say everything went great during meiosis 1. The tetrads separated into 2 cells. Well, shouldn't those now what are called, remember, during meiosis 2, these are called non-homologous sister chromatids. Non-homologous sister chromatids. Shouldn't they separate like this during meiosis 2? The non-homologous sister chromatids separate during meiosis 2? Yes, this is normal. But what if there's nondisjunction? What if they don't separate correctly during meiosis 2? Look at this. What if both of them go into this gamete and 0 goes into this gamete? Well, now when the sperm comes along, or the egg, it doesn't matter, the other gamete comes along. It fertilizes this, fertilizes this, fertilizes this, fertilizes this. These two are going to be normal zygotes. These two are going to be aneuploidy. You're going to have a monosomic, and a trisomic. So this would be 2n plus 1. It has an extra chromosome. This is 2n minus 1. It's missing a chromosome. And these two embryos down here are going to be just fine. And remember I said not only does non-disjunction occur in meiosis 1 and meiosis 2, but it could happen in mitosis, right? So remember during mitosis you also have metaphase and anaphase. Well, what if it doesn't happen correctly and you end up with only one chromosome 1 in one cell and 3 chromosome 1s in another? Well, now you have a 2n plus 1 cell line and a 2n minus 1 cell line. Okay, so that can also happen. Concept check number 5. A diploid organism has 2n equals 36 chromosomes. Okay, 2n equals 36. How many chromosomes will be found in a trisomic member of this species? Well, trisomy means 3, so it would be 37, right? 2n plus 1, 37. So now let's take a quick little break time with Gizmo and Wicket and come back strong to finish up this chapter. Alright, welcome back from break time with Gizmo and Wicket. So what are the downsides of aneuploidy? Again, remember that if you have abnormal gene expression, you have too many copies of various genes that could lead to developmental defects. If you have too few copies of a gene, that can also lead to developmental defects. A lot of times you need the 2 homologous chromosomes with their 2 respective alleles producing the correct amount of gene product, the correct amount of protein in order for everything to run smoothly and in order for development to occur. So again, because of abnormal gene dosage effects, a lot of times aneuploidy interferes with normal development. Not only interferes, but a lot of times with humans, it's just not compatible with life. You can't have, for instance, 3 chromosome 1s and be born. There's just too much of a gene dosage effect on development, and that does not result in a viable human birth. The major exception here is the X chromosome. Remember, you could have 2 X chromosomes. Remember Klinefelter syndrome? 2 X chromosomes, 3 X chromosomes, 4 X chromosomes. You could have, you could be female, so you don't have the Y chromosome. You can have multiple X chromosomes, poly X, and be fine. And remember, the reason for that is because of the silencing, the bar bodies. Remember the silencing effect that occurs with the sex chromosomes. So just because you have 4 X chromosomes doesn't mean all 4 are expressing their genes. All but one of the X chromosomes is silenced. We touched on this previously. Now, in humans, it's really fascinating because for unknown reasons, there's a really high percent chance that humans will conceive a child with chromosome abnormalities. You'll have aneuploidy in a remarkably high percentage of conceptions. So findings suggest about 30% of all conceptions are spontaneously aborted or miscarried, and that's usually due to these aneuploidy events, and that's usually too early in development to even be aware of pregnancy. Chromosome defects are present in at least 50% of spontaneously aborted human fetuses. Usually aneuploidy is the culprit. So many of the miscarriages that humans experience are due to aneuploidy, this incredibly high and unknown reason or event of aneuploidy during conception. Only about 2% of all fetuses with a chromosome defect survive birth. Here's an example in humans. Here's an example. Trisomy 21. Trisomy 21. Remember, trisomy means 3 copies of a chromosome, and this would be chromosome 21. So these are humans that have 3 copies of chromosome 21. This is also known as Down syndrome. It is the most common autosomal aneuploidy in humans, affecting about 1 in 700 human births worldwide. Primary Down syndrome is when it happens kind of randomly because of nondisjunction. Familial Down syndrome is an inherited version, an inheritable version of Down syndrome, and it's due to a Robertsonian translocation. So we'll touch on both of these. Let's start with primary Down syndrome. It accounts for about 92% of those who have Down syndrome. And it's usually due to nondisjunction during egg formation. So the female egg, either in meiosis 1 or in meiosis 2, could have nondisjunction event occur in chromosome 21, and that results in the trisomy when upon fertilization by the sperm. Now, in familial Down syndrome, there's a Robertsonian translocation between 2 different chromosomes, chromosomes 14 and 21. Part of chromosome 21 shuttles over to chromosome 14. So now it's almost like you have 2 chromosome 21s because a big part of chromosome 21 is now attached to chromosome 14. So when you inherit chromosome 14, it's like having an extra chromosome 21. Does that make sense? So you're going to have a lot of the same phenotypes of chromosome 21. Individuals have 46 chromosomes, but have an extra copy of part of chromosome 21, which is attached to chromosome 14 due to a translocation. Well, we'll get to this in just a second. But first, let's take a look at the heritable or the non-heritable version of Down syndrome, primary Down syndrome. So in primary Down syndrome, again, this is usually caused by a non-disjunction event in the mother, in the egg, and this results in what? It results in 3, notice here the chromosome 21, there's 3 chromosome 21s, right? There's 2 sets of chromosomes. However, with chromosome 21, there's actually 3 chromosome 21s. So this would be a 2n plus 1 situation. So a total of 47 chromosomes, 3 chromosome 21s. Now, here, this is an example of what you would see with a Robertsonian translocation. A Robertsonian translocation. So in this case, you have translocation of chromosome 21 onto another chromosome, resulting in familial Down syndrome. 21, part of it was transferred onto chromosome 14, and so you would have the same effects there, and it would be heritable. But there's other versions of aneuploidy in humans as well. Trisomy 18, which affects 1 in 8,000 live births. This is also known as Edwards syndrome. Trisomy 13, Patau syndrome, 1 in 15,000 live births. And trisomy 8, 1 in 25,000 to 1 in 50,000 live births as well. Now, here's a question. Why is there a drastic decrease in the frequency of trisomic syndrome from chromosome 18 to 8. What do you think? If look at this. This is a very good question. How come there's 1 in 700 humans with trisomy 21, 1 in 8,000 for trisomy 18, 1 in 15,000 for 13, 1 in 50,000 for 8? Why do you think the numbers keep getting more and more rare? Why do you think it gets more and more rare by the time you get to trisomy 8, do you think? Take a look here. Look at the human karyotype. Why would you think that it's much more common to have trisomy 21 than, let's say, trisomy 8? Well, if you're thinking what I'm thinking, the answer is a lot simpler than you would, anticipate, and that is because it's just longer, right? Look, chromosome 8's longer, chromosome 7's even longer, 6 is even longer, chromosome 1's the longest. So if it's the longest, doesn't that mean most likely it harbors the most genes? You guys follow me? Chromosome 1 harbors thousands of genes. So if you duplicated it, now you have thousands of extra genes, thousands of extra gene products, that means proteins, and thousands of opportunities to mess up development. And same thing with chromosome 2. You've got thousands of genes, which make thousands of extra gene product, thousands of opportunities to mess up fetal development. You see, so you're not going to find a human with 3 chromosome 1s or 3 chromosome 2s, because having 3 copies of those chromosomes is going to cause such a disaster during development, fetal development, that you're not even going to get a viable birth, because again, dosage effect, right? Dosage effect of the genes. The smaller the chromosome is, look at chromosome 1, it's this itty bitty little chromosome. See, chromosome 21 is this itty bitty little chromosome. It's much less likely to have, as many deleterious genes on there, genes that are required for certain dosage, and there's just so few genes on there in general. There's so few genes, it's much more likely that you could get away with having an extra one. Does that make sense? So that's just a very simple explanation here, why it would be more common to see trisomy of some of these shorter chromosomes than some of these longer chromosomes. Does that make sense? It all feeds back to dosage effect. You put the wrong dosage of genes in the cell, you're going to get the wrong dosage of proteins that come from those genes, and you're going to mess up development, right? So because development requires the right dosage of genes. So concept check number 6. Why are sex chromosome aneuploids more common than autosomal aneuploids in humans and other mammals? Well, we touched on this, didn't we? You can have sex chromosome aneuploids. You can have 3 X chromosomes, 4 X chromosomes. You could have all kinds of X chromosomes, and that's not really going to affect you, because remember, all of the X chromosomes get silenced except 1. So because of that, you're not going to have deleterious effects. But the autosomal chromosomes, you don't silence extra copies of. And that's going to lead to dosage effect, right? Now, here's another problem with aneuploids. Aneuploidy is responsible, well, maybe that's a little strong of a term, but aneuploidy is seen and heavily associated with many different cancers. There are many different cancer cells where you often and consistently see chromosomal aneuploidy. You see extra chromosomes or missing chromosomes. So it's not uncommon at all to see aneuploidy in cancer cells and tumors. And aneuploidy can change, the frequency of aneuploidy can change with age. I can show you this as well. This is very fascinating stuff. So why is there a change in aneuploidy over time? Let me show you something. This is a very fascinating little graph here. Look at this. This is very fascinating. What you're seeing here is the incidence of primary Down syndrome and other aneuploidies, that how they increase with maternal age. So this is the age of the mom, right? The mom in her 20s, 30s, 40s, 50s, right? If you have a child in your 20s, you have approximately 1 in 2,000 chance of having a child with Down syndrome per 1,000 births, right? This is per 1,000 births. So you have 1 in 2,000 chance approximately of having a child with Down syndrome. In your 30s, this drops to, or rises, I should say, to about 1 in 900 or more. 40s, 1 in 100, and by, late 40s, 50s, 1 in 12 mothers give rise to child with Down syndrome. Isn't this fascinating? It's like logarithmic, right? So why is that? What is the possible interpretation of this connection? Well, we don't know for sure, right? Geneticists don't know for sure. Female mammals are born with primary oocytes suspended in prophase 1 of meiosis. So what this means is the way the gametes develop into the way the gametes form in males and females is different. Does that make sense? The way the sperm forms is different than the way the eggs form. Males are producing sperm all the time, and they're producing sperm from the germ cell to the sperm cell all the time. Does that make sense? Spermatogonia are doing spermatogenesis all the time, and you're getting sperm produced by the millions all of the time. But with females, you may have heard the expression that you're born with all the eggs that you're gonna use. And that's true. When a female mammal is born, and humans are mammals, a female mammal is born, she's born with primary oocytes. Remember, we talked about what primary oocytes are in a previous chapter. Primary oocytes that are suspended in prophase 1 of meiosis. So you're born with X number of oocytes. If you're a female, you're born with X number of oocytes. These are suspended in prophase 1 of meiosis. They're just there in prophase 1 of meiosis, ready to, complete meiosis and make an egg for fertilization. Just before ovulation, meiosis resumes. Then you get the first meiotic division, and then you do the to produce the secondary oocyte, and then at this point you have to wait until fertilization by the sperm. Fertilization by the sperm. Only upon fertilization by the sperm do you undergo the second meiotic division to form the zygote. So what they're thinking is, well, by your 20s, you've waited about 20 years for this primary oocyte, to begin the process of meiosis and do the first meiosis division. By your 30s, you've waited 30 years. By your 40s, you've waited 40 years. And what they're thinking is maybe components of the spindle, the mitotic spindle, or the kinetochore, or part of the centromere, some part of the spindle and other structures required for chromosome segregation, during anaphase, metaphase, anaphase, telophase. Some of these these delicate components might break down during the long arrest of meiosis, waiting until you're 40 for that cell division to happen. Maybe some parts of the spindle aren't functioning correctly by that point in time. So that's the best hypothesis there is right now. And there's some evidence to suggest that. But again, we don't know for certain. But we do know for certain this. We know that the chances of mothers being born or having children be born with Down syndrome increases almost exponentially into the 50s here. So very interesting stuff. And again, why wouldn't this affect the males as much? Again, probably because Males are making sperm from scratch all the time. These cells haven't waited 40, 50 years to undergo meiosis 1 and meiosis 2. These cells are being regenerated from stem cells in the gonads all the time, right? That's the hypothesis. Very interesting stuff, though. Now, polyploidy, we didn't touch on this much, but polyploidy is when you have a whole extra set, not just one extra chromosome or one missing chromosome, but a whole extra set of chromosomes. So instead of 2N, now you're 3N or 4N or 5N. And it's a major mechanism by which new plants evolve. You know, plants, when you're talking about polyploidy, a lot of times you're talking about plants, because many, many plants are polyploid. Look at this. I don't need you to know a ton about polyploidy because this isn't really a focused plant genetics class, but just take a look here. Potato is 4N, you know, a banana is 3N, a sweet potato is 6N for some reason. But look at this is my favorite, strawberries, 8N, 8N, you know, sugarcane, 8N. So very fascinating stuff. And so you frequently see polyploidy in plants. Very interesting stuff. And this here is just a table of review terms that we've covered. Otherwise, we've reached the end of the chapter. Very interesting stuff, isn't it? I think, you know, it really makes all the concepts come together once you look at the chromosomes and you look at human diseases and you start relating the mechanisms behind some of these human diseases. And you could trace them back to these types of mutations and really understand how they work. I think it's fascinating. But anyway, leave any questions you have in the comment box below. It's been fun, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.